I would like to just invite Jeff Leek up. Jeff is a very long-time contributor to the project. He's now the head of AI-based technologies at the Fred Hutch. I think he has a lot to share with us about this, and I'm just going to, without further ado, invite him up to the talk. Thank you.
Thanks, Vince. I really appreciate it. Yeah, so I do want to say thanks to Vince and to the organizing committee, and I know everyone that was involved with this. These always take a lot of work. Bioconductor is very near and dear to my heart because when I, was a baby computational biologist, the bioconductor community really kind of took me in, and like basically my entire sort of mentorship chain going up through my career has been people in the BioC community. People have really helped out Vince, but also people like Rafael Rosari, who is basically my main faculty mentor, and Giovanni Parmigiani, who also have been involved in the project. The thing I thought I'd talk about a little bit today is, I've been working in this field for a little while, and I think one of the things that's so amazing about Bioconductor is the way that it enables a big community of people to work on hard problems in science. And it does that through the use of software and data technologies. And one of the reasons why I took a job like I took here, which is professor and chief data officer, is to see if I can kind of take some of that ethos and bake it into what happens in a cancer center and dealing with patient data, which has a lot more requirements around it than some of the data that we deal with in a public way. So first I want to welcome you all to Seattle. Thanks for coming and being here. Really appreciate it. Exciting to have it back here in town. And as I showed just a minute ago, I go fast and use a lot of slides. So if you want to follow along, you can find my slides at this website. I'm going to declare a couple of conflicts of interest up front, and the main one to pay attention to is the second one, because I'm going to talk a little bit about synthesized bio at the end of my talk, and I'm a co-founder there, so you should take everything with a grain of salt when I talk about it. Okay, so there's this, I grew up in Idaho, and there's this famous phrase, Idaho, it's not just potatoes, but mostly. And so I kind of have taken that to heart when I think about AI. Like AI, it's not just LLMs, but mostly today, and so I wanted to talk a little bit, like zoom out a little bit and talk about what I think AI can really do in the healthcare space. And I have a couple of different seats from where I'm looking at this problem. On the one hand, I'm a professor and I do research and work in Bioconductor, maintain Bioconductor packages. On another, I'm a chief data officer and run kind of a data ecosystem at the Fred Hutch. And then on the third, I'm a co-founder and co-CEO of a startup company. And so I think that there are kind of four big areas where I think there is actually a lot of potential for AI, where you define that quite broadly to have an impact on the field. Care and operations, drug discovery and development is obviously a big one, and then consumer health is another place where we're seeing it be deployed a lot, but we really need to think about each of these. So one of the things I get asked to talk about AI a lot, and one of the challenges about talking about AI is that like everybody has a different definition of AI, and so it's a little hard to even know what we're talking about when we're talking about AI. I like to use a really simple definition. To me, an AI is any system that has these three parts. It has a data set, it has an algorithm trained on that data set, and it has an interface that humans can interact with. That could be as simple as a very small set of like the iris data in our linear regression model and a Shiny app. That to me, I know it's ridiculous, but that's AI to me, because it's like you interacting with a system. It can be as complex as some of these multi-billion-dollar systems that are being built, where you like take a bunch of data that's text on the internet, download all of Reddit, chop it up into little pieces, stuff it into a very complex model architecture like a transformer architecture, and put a chatbot interface on it. But there's lots of other ways that you can imagine doing this. And for me, anything that gets has these kind of components is an AI. And so when you think about it in that context, AI does have a lot of potential to impact things, not just in the LLM space, but in lots of different spaces in biomedicine. And the reason this is forever fresh kind of a topic for the course of my career are these two graphs. This graph, if you haven't seen this graph and you're in Bioconductor, you must have like bumped your head or something. Like the cost of sequencing a human genome has gone from $100 million when I was a grad student to like less than $1,000 today. That is a pretty dramatic change in economic costs just in the course of my career. At the same time, you can rent more computers than you could ever before, and there's lots of politics involved in that, and I'm not going to talk about the politics of data centers here today, but I will say that there is only more computing coming at us over the next decade, and so how are we going to use that computing to answer important problems? And these two things, when put together, allow you to build big, complex models that do cool stuff that people are excited about. So it feels like this all the time in the intersection of sort of data and biomedicine. So one question I get asked a lot, including by, various leaders of various different institutions where I sit on advisory committees is, aren't the big AI companies doing all the AI stuff? Like, what's the point of us even doing AI? Like, Anthropic and OpenAI are doing all the AI for us. And so should we just shut it down? Should we shut down all of biomedical science? Should we just shut it down because it's all going to get done by models? And I think that's, obviously in the ether, those kinds of ideas, but I think there's a real reason that that's never, in my opinion, going to be kind of the long-term, or at least not for a long period of time, is going to be the long-term way of the world. And the reason is there's very distinct things that an AI company and like a healthcare system or healthcare researchers have advantages with. So an AI company can focus on a very specific model. They can move much faster than we can move, and they can tolerate risks that we would never tolerate in biomedical science. And so that lets them do things, a certain class of things very well. What we have as a healthcare center or as people who work in biomedicine are decades-long development of patient trust, a huge amount of data that we've collected in painstaking ways over periods of times, and the experience of people like you in the room who actually know how to do stuff with this data. And so when I think about what do we do in AI, what do companies do in AI, this kind of framing helps me think about which parts are the parts we do. So I wanted to start with like one of these four challenges, consumer health, and talk a little bit about how, even though there's this acceleration of AI models, we still have a lot we can do. So this is a project that I worked on with a graduate student who's leading this project here. He's at UW and Fred Hutch, Yimin Zhao, and he and a bunch of clinical fellows have been working on this project where we're trying to figure out, do LLMs answer patient questions well? That's the simple question that we're trying to answer. So what we did is we worked with the Pancreatic Cancer Action Network, which is a pancreatic cancer foundation. They have a helpline that you can call in when your loved one has been diagnosed with pancreatic cancer. We abstracted many, many patient questions and anonymized them from these phone conversations that were had by people from the Pancreatic Cancer Action Network. And then we had a bunch of LLMs answer those questions. So we just basically took the question, gave it to an LLM, got the answer. And then we had clinical fellows who are experts in this disease develop a rubric that was essentially like, for this question, it must have these things in the answer, it must not say these things, et cetera. And then we applied that rubric to all of the answers from all of the LLMs and gave them all a score. And so one of the things that's interesting is you can kind of look at these graphs here. These are some of the results. And so on the top graph, it's just the fraction of the rubric questions that the LLMs got right. And so you can see some of them do reasonably well, like up to almost 80% of the rubric questions get right by the most advanced frontier models from someplace like OpenAI or from Google. Some of them don't do as well or didn't do as well in this evaluation. And in particular, the open source models were doing a little bit worse. But the more important thing and the more interesting thing, I think, to me is we also just evaluated how many times it had a factual error, like tell a patient the wrong thing. And it turns out that there's a fair number of times where even if they're kind of getting most of the rubric right, they're giving a patient a factual error in their sort of clinical diagnosis. And so even though these models are very powerful and seem like they'll tell you the truth, they often have errors in them. And part of the reason for that is the training data for these models is largely Reddit. And so if you get the right answer from Reddit, you'll get the right answer from an LLM. But generally what this means is we, as the biomedical community, can really have a big impact on consumer health by helping these models get better at the things that they need to get better at for patient health. So despite this kind of, hiccup, why am I inspired in this moment? And I think one of the things that inspires me about this moment in time is something that we learned from Bioconductor and from the projects that are around Bioconductor, which is if you make data available in a safe and secure way to a lot of smart people, amazing things happen. And the key is the more smart people that can do that, the better the outcomes that you have. There's a pretty direct correlation between those two things. And so I'm going to talk a little bit about how we're doing that in the context of the Fred Hutch and then at national scale and what the impact of it is. And really this is thinking a lot about this end of the spectrum now, care and operations. as the place where you're thinking about using these data. And the kind of related idea that I've worked on in my past career is something called the Recount Project. So this is a project that started when I was at Hopkins with a couple of other faculty members there. It's a bioconductor package. It's used quite widely. And all it does is it takes most of the human gene expression data that's posted online, it processes it under a common data processing pipeline, removes some of the artifacts, and like makes it available for people to easily download it through a bioconductor package. So it's just like simplification of how do you get access to these data. And then it also like layers on some predicted metadata so it's a little bit easier to use. And so this kind of project is awesome because we made that resource and we published it. And then a few years later, this Nature paper came out where this other group with none of the same authors as the Recount Project published a paper in Nature where they completely reannotated the human transcriptome for both research and clinical purposes. They used our gene expression data that we had published as the way that they could identify where there were new transcripts that hadn't been identified previously. And so this is the example of this resource existed. People I didn't even know who were working on a problem I didn't even know they were working on somewhere else had a major impact on the field because this resource was available. So this has kind of driven my thinking and our thinking about what we want to do here. And a sort of second inspirational example for this is a former colleague of mine at JHU named Suchi Saria. So Suchi took ambiently available clinical data from the ICU. So if you're in at the hospital, people collect a lot of information from you, your temperature, your blood pressure, a bunch of other labs that they could take from you regularly. And she went and looked, could I build a predictor of, are you going to have a hospital-acquired infection before the doctors diagnose it? So can I predict if you're going to get sepsis? It's a hospital-acquired infection. And she built this model, and it was amazing. It worked really well. It was in the news everywhere. It was a really big deal at the time that it came out. And then for a long time, there wasn't a lot more from Suchi's group about this project. It was like such a inspirational moment, and everybody was very excited about it. And then like, all quiet for a while. And so about 7 years later, she came out with this tour de force set of papers. And if you care about translational biomedical data science, these are the papers every student should be required to read. And so what she did was she took her model, she engineered it into the Johns Hopkins electronic health record system. That's a big hospital system. She ran a human-computer interaction study to study the way that physicians used the alerts that it gave them or didn't use the alerts that it gave them. She ran a prospective randomized study to show that it actually reduced morbidity and mortality if they used this alert score. And then she basically studied the factors that would involve like how to make that score more effective. And so this is an unbelievable tour de force of papers. She then founded a company. That company's very successful. Things are going really great. And the thing that was inspirational to me about this, and the reason I actually took the job at Fred Hutch as a chief data officer, was the two dates, 2015, 2022. Why did it take 7 years? Suchi's very smart, and she works very quickly. It should not take 7 years. It takes 7 years because the systems that enable you to do this kind of work are very complex. They're very hard to navigate, and they are often at this weird intersection of legal, technology, governance, compliance, ethics, et cetera, that makes it very hard for you to just like take a model and deploy it someplace else. And so the same principles applied to Suchi that applied to Recount, which is she had access to this ambient data that was collected. She could build these models. Those models had high impact. That impact also requires you to understand, again, going back to the definition of AI as data, algorithm, and interface, you have to understand how people are going to interact with your algorithm. So she did this study, and she studied all the physicians that were using these using these alerts from her model. And she found interesting things, some of which are pretty intuitive, like if you give a high alert, people will pay more attention. Some of which are also intuitive. Some providers care about alerts and some don't. And then some that are also really interesting, but maybe wouldn't have been intuitive, which is like at certain times of the day, people pay attention to alerts, and at certain times of the day, they don't pay attention to alerts. So like if you deliver the alert at the wrong time of day, it might not work. Even if the model worked, even if it was right, even if it was delivered to the right person, it still might not work. So it's really interesting that like even when you have a working machine learning model, it needs the human element. And then the last piece is, and I think this goes to some of the points Vince was making a minute ago, which is she studied, do physicians trust these alerts or do they not trust the alerts? Because they didn't understand the model. The model was a very complex machine learning model, so it was not like an interpretable model by any means. So how do they trust it? they have to trust it by essentially building a relationship with these alerts. Like, did the alert work when I used it last time, or did it waste my time? Did the alert annoy me, or was it good? And so like, you can build relationships between a human and a computer that allow these things to be very effective, but it takes work. And so our thought was, when I came here, was how do we build a translational patient data ecosystem that allows democratized access, just like we did in genomics? And so the first thing that I did when I got here was I hired this person, Sean Cross, who's also been involved in the bioconductor community. And Sean went out and asked everybody, I'm new chief data officer, what do you need from data at the Fred Hutch? And this is my approach to figuring out new problems, is like, let's go try it out, see what happens. And so Sean went and did that and went and knocked on a bunch of people's doors. And what we found is what the large majority of people wanted was access to the clinical data in a way that was safe, so that they knew they weren't doing something weird with the patient data. They wanted a way to have support to analyze that data, and they wanted infrastructure on where to analyze that data. Those are like very consistently the themes across thousands of people around the Fred Hutch. And the way to solve this problem has kind of been figured out before, and so we kind of, this isn't a clever new idea, it's an idea that we've adopted, but the first articulation of this idea that I've seen is this person, Andre Karpathy, who at the time was a director of AI at Tesla. I think he's at Anthropic now. And he talked about this idea of an AI engine. So often in biomedical science, often in genomics, we do a study and we collect data, point in time, we analyze that data and we report results, right? Like that's the most common thing. You don't like go back and collect more data from those same patients adaptively based on the data you collected from the first experiment, then do that again, then do that again, then do that again. The places where AI is really advancing quickly are places where you can do that. So he was talking about cars in this case, and you have a Tesla car, it collects some data, that data is then cleaned and put and labeled and put in a place where you can use it. You build some more models, you deploy them back to the car, you collect more images, and you keep doing that cycle over and over and over again until things get better. And so in biomedicine, we haven't really had systems like that where you can do this kind of like iterative improvement. And so we've been thinking about how do you do that in the context of a healthcare system. And the real answer is you can build these kinds of engines. They've had lots of names over the course of the years, but learning healthcare system is a very famous term you've probably heard, but there's a bunch of others. And the idea really is you collect data from your patients, you build new technologies, whether it's new treatments, new decision support tools, et cetera. You run clinical studies to validate them, you deploy them back into the patients, and then you repeat that cycle. Famously, the Fred Hutch has a very shining example of this, which is bone marrow transplantation. So bone marrow transplantation is, if you don't know about this, it's basically Control-Alt-Delete for your immune system. And so that's an Amy Pagarigan joke that I borrow. But the idea is really that you can, basically irradiate somebody, remove their immune system, and then give them a transplant, and then that transplant will rebuild their immune system. That idea was discovered here, and it was discovered because, and kind of validated here because of a way that they were collecting data about every patient that they got here, and they could tune those kind of those treatments to those patients and improve them iteratively over time. And so our question was like, can you build that at scale? And the challenge of building a system like that at scale is like, I'm a nerd, and software and data is where I would like to spend all of my time, but like that's like 4% of building a system like this. Most of it is trying to make sure that the right people have the right data, making sure that all the legal stuff is in place, making sure you have the right agreements with all the right people, making sure that everybody agrees to how things are supposed to be working, making sure nobody's doing something creepy with the data, et cetera, et cetera. And so it turns out that these kinds of problems are actually quite complex, and that they involve all these different components. One of the lessons I've learned here is that data is political. It's deeply political. When you take a job like chief data officer, you think you're going to spend all your day talking to nerds building systems, and what you actually have signed up for is to be a politician and are glad-handing and kissing babies and shaking hands. And so the question is, like, how do you actually get people excited about these sorts of things and get them involved in them? Because you have multiple groups that have to be involved. The first thing we had to do was get some data. And so for us, we have a relationship with the University of Washington. University of Washington hosts our EPIC, our electronic healthcare record system. This is a common arrangement where Vince is from, has a similar arrangement with a big hospital. And so we had to set up a system where we could decide which data we were going to pass back and forth, set that up into kind of an ecosystem of like, how do we actually turn that into code? And then how do we get everybody to approve it? And then how do we share the data over? This is a very complex process, and Amy Pagarigan and her team in data governance here at the Fred Hutch have really set up this system for basically allowing us to create secure data transfers between all these different orgs in the Fred Hutch. The second thing we learned is that enterprise software requires collaboration and investment. We have now built this system called Cards here that hosts all of our multimodal patient data. It links all the patient data about our patients in one secure system. It takes it in from many, many different sources, whether that's for-profit companies, whether that's from electronic healthcare systems, whether it's from other institutions like the University of Washington. And it stores it all in one system, and then it gets turned into a bunch of different data products that people can use to do their work. And when you do that, that's requires work with information security department, with the IT department, with the data department, with the legal department, et cetera, finance department, et cetera, et cetera. And then you want to make it available. So you have to build systems that make it available to people. And so we have a registry of all the patients that we have information on in our systems, securely stored in our systems. We have to set it up so that it updates every single day. So like we collect more data about patients every day. We have to get it so that there's a steady stream of data that's coming in. This is all in service of building that engine. And then you have to make sure people only get the data that they need. So like one of the challenges is you can't just, unlike some of these like public access data sets, I can't just like dump this data on the internet and say you can all have it. It's like this information is health information about patients. We have to be very careful with it. We have to be careful about who gets access to it. Do they have the right approvals, et cetera. So you have to build a whole system for essentially like figuring out who gets access to what data at what time and do they have all the right approvals and then monitoring what data that they have. Another big component of making it available is structuring it. So like you saw there were some efforts from Bioconductor to structure genomic data, which is certainly true and standards are so critical. We here have been having to adopt, open standards for biomedical data and de-identify and structure the data so that people can have access to it in that way as well. And then one of the things that is a huge culture change, but we're trying really hard to be able to do this, is make this data available as self-service. There's been this sort of traditional approach to how biomedical data has worked, and maybe even in the old days how genomics worked, where you could only really get to genomic data if you went through a specific analyst, if you went through a specific person who had access to it. And so one of the things we want to do is figure out ways to de-identify data, make sure that none of the information that we don't want out there to be available is available, but allow people to go say, how many patients do we have that we've given a bone marrow transplantation to? How do we know if we have enough people to run a clinical trial on those patients? And so building the tools that make that possible. So what this enables is that people can bring their own data and link it with the existing patient data that we have here, which creates this growing resource that's more and more complete. The inspiration for this idea is actually another project that was both started and led here out of the Fred Hutch called the Women's Health Initiative, which is a major project that has followed a group of women since a clinical trial ran about 30 years ago and has followed them for like 30 years and collected incredible amounts of information about them. And people can use that resource to learn a lot about women's health. It's an unbelievable resource. And if you take some of the samples and you do genomics, say, on some of those samples, you have to contribute the data back to the resource. So there's this continuous growth of the Women's Health Initiative resource. We have a similar model for how we're building patient data here. And then it allows you to do data analysis in R, Python, Bioconductor online so that you don't have to export the data and we don't have little pieces of patient data living all over the place. It also enables the safe use of LLMs and that kind of AI. Today, it's a little concerning what you should absolutely not do is take all your patient data and upload it to Anthropic or OpenAI. So you want a way to securely protected use those models, but not have it fly out of your systems. And so you can do that with these kinds of things that we built. And then you can pick which model that you want to use. So this is kind of an incredible resource that's being built up, that's being used by people around here. But the key for any resource is building a pit of success. So this is another phrase that I've lifted from a person named Hadley Wickham, who's an R programmer. So he had this idea of a pit of success, which is you can make it really hard to do the right thing. It's like climbing a mountain. or you can make it really easy to do the right thing. You're walking along and you just fall into the pit. And so I think what we've been trying to do is think about ways that we make the easy thing to do the right thing to do the easy thing to do. And so building pits of success is a huge part of making these kinds of ecosystems and these kinds of systems something that everybody can use. And so when we think about how Fred Hutch people are using our systems, if they accidentally step in the wrong place and they're going to do something wrong with the patient data, we want systems that will catch them and make sure that they don't land on their head and that things go well for them. And so that's partially human systems that you have to build, like the things like the data sort of governance things that I talked about. It's partially the technological controls that we've built. And it's partially these sort of ways in which the data are processed and normalized so that you're not giving people data that are on their surface kind of radioactive or hard to deal with. More importantly, one of the things that's so important, and I think Bioconductor has also inspired and something that I think we've kind of learned from and we're doing here is giving people an opportunity to learn how to serve themselves. One of the things that's so amazing about the Bioconductor project is it's created this software ecosystem where any grad student can go learn how to do bioconductor stuff and start analyzing big data sets. And that, when I started as a grad student, that was not the vibe of how data analysis was done. It was like very constrained to people who were like PhDs in biostatistics and PhDs in computational biology, and they kind of like kept it tight to that group, and that was, you know, there was this sort of like gatekeeping of analysis. And then Bioconductor, I think, was one of the first kind of really big efforts to like change that dynamic and say, if you're a grad student and you're generating some data, you should be able to analyze your own data and you should be able to figure that out. And they do that with training and support. We do that here as well. Ted, who has helped co-organize Bioconductor, is sitting here in the front of the audience, leads our training group, and Chris is also here, is also leading our training group together. And they do a bunch of training for people to learn how to use all these tools. And then we have what's called data house calls, where it's like, call a friend who is a data analysis expert for 30 minutes and get some information from them about how to solve your problem. And so really the goal here is this graph that I made like a long time ago. This is, you have these kind of steps in a data analysis. And there's kind of two ways you can think about how you move through this. One is you get in line for help doing, you know, the first step, accessing the data. You get a person, they help you. Now you've got some data. Now you process the data. You could go back and get in the back of the line and wait for another person who can help you process it. And then you can do the next step and the next step. That's very slow. One way to skip that line is if you know how to access the data on your own, you could just do it on your own. You don't have to wait in that line. So that's like one line you get to skip. If you know how to process the data yourself, you get to skip that line. This is such a critical piece of like systems thinking about how to build data science systems that work for institution scale, because we will never have enough data scientists and data analysts we can hire. Even with all the AI coding tools, even with all of that, the demand for people who know how to analyze data outstrips the number of people we have to analyze it. And so you have to think about ways that you can help people kind of get past those roadblocks that they run into. The other thing that we've learned from Bioconductor is that software is a great democratizer of that exact kind of component. And so like, even if you train somebody to how to do something, if the software isn't very good, they can get stuck. And so we've been thinking a lot about how do we do this in the space of AI. And one of the ways that we've been doing this is a joint work with a former postdoc who's now a faculty member at WashU, is building this software called Scorcher. Scorcher is basically a piece of software that tries to make it easier to make machine learning models. So if you actually want to build a machine learning model with something like PyTorch, which is kind of like the most common software for building these big complex machine learning models, you have to do a bunch of different steps, many of which are just technical and aren't actually like related to the problem you're trying to solve. Like creating a data loader is like creating a very specific, you can think of it as an S3 class. It's like an object that you have to create a very specific way in Python. If you don't know how to make that, you can't make a machine learning model, even if you know all the other stuff. So one of the things we've been trying to think about is how do we make really simple software that lets people do things like use one command to take a data set and create a data loader, then pipe that into something where you add a bunch of layers to a neural network. Then pipe that into something where you compile that model into an object that can actually be trained on like a cloud or something like that. Then you fit that model by like sending it to a piece of computational infrastructure and actually have it fit. And like, rather than this being like hundreds and hundreds and hundreds of lines of code, it's like 5 or 6 lines of code where you just have each of the steps spelled out. So this is an example that Steve came up with. This is a generative model. This is a diffusion model. If you've ever played with any of these models that generate images, like with Nano Banana or whatever, they're diffusion models. This diffusion model is just to create dinosaurs. And so from these point clouds. And on the left is the Python code that would take you to write that in PyTorch. And on the right is the Scorcher code that you would take to basically write that exact same model and generate those dinosaurs. And so one of the things that's so interesting about the way that Bioconductor and in this case Steve have been thinking about these problems is they abstract away, these kinds of packages abstract away all the stuff you actually don't need to know how to do because it's just some technical thing, and it just leaves the part that is actually the important part when you're building these pieces of software. And so this allows us to kind of build much more complex models and have more people build more complex models, so it's not just a few machine learning people. So that's what we were kind of building at the Hutch, and it's exciting because that enables a certain class of people here. But then we were sort of thinking about, okay, can we do this at a bigger scale? And I'm till 10:30, right? Yes, okay. So you check your watch, so I start freaking out. So the next step was, okay, we're doing this at Fred Hutch, very exciting. Can we do it beyond the Fred Hutch? We aren't the only ones building a platform like this. So like the Fred Hutch is building one like this, but this again is not like rocket science. Like a lot of people have thought, hey, maybe we should get all the patient data together. And so they're being built in lots of other places. And these platforms have shown over and over again that they have big impact. These are two that come from a colleague, two papers that come from a colleague at Memorial Sloan Kettering in New York. One where they're basically looking at non-small cell lung cancer and trying to identify predictors of response to a particular therapy. And another where they're trying to replace a very common breast cancer screening test that costs $3,000 or $5,000 with just a AI model that takes your pathology readout and predicts your breast cancer recurrence risk without having to do that $3,000 or $5,000 test and wait lots of time. Both of these are built with their equivalent system at MSK. All this to say, Fred Hutch is doing this. We're not the only one doing this. What if we wanted to do this at scale? One of the challenges is that even though all of us are building these systems, they have to be secure, they have to be private, they have to be carefully managed. And so what that's creating is these silos, like everybody's building their own. They're building it on their own system. They're building it on their own platform, which means if I build if my colleague at MSK builds a model for breast cancer recurrence risk and I want to deploy it here, like my first step is a bunch of data engineering. Then I can deploy his model to my system. Then I can maybe test his model. So like the passing back and forth of these models is basically not happening very much. There's also hard to generalize, like the patient population that's seen by people at my former institution, Johns Hopkins, is very different than the patient population seen here at the Fred Hutch. So like you might build a model that works great at the Fred Hutch and it doesn't work at all in Baltimore. And then there's all the like technical complications. They're on different clouds, different data models, different regulatory systems. They have different requirements. It's just kind of a mess. So what we wondered, is there a way to connect all these data nationally? Is there a way to actually make it possible for all these different patient systems to talk to each other so that you could build and ultimately deploy models much more efficiently? Again, not the first people to think of this idea. I've been involved in, I would say, at least 3 major government-funded initiatives to try to do something like this, and they have all failed because legally it's very hard to do this. It is unbelievably hard to, like, we can't just take all our data from the Fred Hutch and dump it in some other database that somebody else controls. That would be bad for our legal exposure and bad for our patients' privacy, et cetera. So there's been this kind of track record of people being like, okay, let's build a database and dump all the data together. They get about a year or 2 years into that process. The lawyers say, like, there is literally no way you're doing that. And then that's the end of the project. It's happened a few times. So we had this idea of like, what if we did it with federated learning, where the data actually never ever left any of the healthcare systems, and you only actually connected the models between them. And so this idea of federated learning basically says, instead of sending data back and forth, we're going to send code back and forth and summaries that come out from that code. And so, for example, if you want to calculate the average height of all the patients at the 4 cancer centers, one way to do that is you just dump all the patient data in one database and average the heights. Very easy technologically. The lawyers will come after you. The other way is we send a little code to each cancer center. We say, calculate your average height for your patients and the number of patients you used for that calculation. Send me those 2 numbers. And with those 2 numbers, I can calculate the average height for all the patients in the population. I never had to see your actual data. So you can do this for very simple things like that average height. You can do it for incredibly complex things like fine-tuning large language models. So one of the big challenges is the data is different at all these different places. So we had to figure out a way to get every different cancer center to agree to a data model and then map all their data to the data model, which turns out is a non-trivial challenge. But we managed to do that for this project. And really, it's not about technology and data. It's actually a whole collection of steps. First, you have to like get anybody to trust that you're not just trying to steal all their data. Then you have to like set up a right legal and regulatory framework, set up a common data model, set up a cloud platform, and then really on top of all of that, you can start doing cool science. And this really requires trust. So this Kaya project, it's called the Cancer AI Alliance, that is the project that's pushing this, is really, I call it a trust-based exercise. And it started, you can see here, this is the Google Doc where it all started about a year and a half ago. Literally, it was just a Google Talk that I sent to a couple people, including a person who's commenting here, Saurabh Shah, who's the head of computational medicine at Memorial Sloan in New York. And we were like, let's write out some, if we were gonna try to do this crazy thing, let's write out some principles for things we would want, like the data never leaves. People can preserve their IP if they're building cool models. They can collaborate. There's ethical and regulatory compliance. There's transparency and accountability. And so we built this kind of set of principles that have carried this project through. And then we went out and raised a bunch of money to try to sort of build this prototype of this federated learning system. And in the first year of that project, we created a collaboration agreement between the 4 cancer centers that allows us to do this federated learning. We set up an expedited IRB pathway. You have to get IRB approval to do these kinds of studies. And we set up, now you got to get 4 times an IRB approval because there's 4 different places involved. So you got to set up an expedited way for that. We mapped 1 million patients' data, longitudinal data, de-identified it, and made it available for federated learning. We built a multi-cloud platform because everybody's on a different cloud, so you have to be able to send these code back and forth. And then we started our first 8 pilot use cases to sort of scale this idea up. What Kaya really is a shared resource. And so shared resources are things that nobody ever wants to invest in and always produce awesome results. It's like a law of nature. So 2 examples of that, one here locally, the protein data bank is this sort of set of protein sequences and then their structures. And it's just an open resource of protein sequences and structures. And then ImageNet, which was made by this person, Fei-Fei Li, is basically just a bunch of pictures from the internet labeled by grad students at first and then later Mechanical Turk. So it's just like labeled pictures. And Kaya is like that. It's more complicated because we're dealing with patient data and we have to be very careful, but it's like the same idea, make an open resource. Why does this work? Like the track record is ridiculous for these kind of resources. So Protein Data Bank led to, here in town, David Baker won the Nobel Prize in chemistry in 2024 for his prediction of protein sequence structure from sequence that was built on the Protein Data Bank. It ended up being the basis of AlphaFold, which is this big Google product that's being used by tons of people. And now there's like 1 million companies being built based on those kind of models. ImageNet is even, believe it or not, even bigger impact. Also, somebody won a Nobel Prize for what they did with AlexNet, which was basically like this big model that predicted the labels of those images from the images themselves. And basically the whole modern AI industry was in some way touched by this. Like almost everybody that's involved in the modern AI industry can trace some roots back to this ImageNet project. So I made this up, but Fei-Fei Li's law is like there's a 30 to 100 times return on investment for any open data resource that you make. These are just some examples of them, but there are a bunch of these out there. And so we're really trying to construct one in this Cancer AI Alliance framework. And so this is already showing impact. This project has moved at light speed, largely because all of the major technology companies in North America are donating to it. So it's moving pretty quick. So we have 3 kind of big things that we're already showing. One is, I'm going to show you on the next slide, this little tool that you can use that accelerates science. We're showing that with the bigger collections of patient populations, you can build models that are much more predictive of, say, things like clinical outcomes across a wide variety of cancers. And then even for like things that aren't really AI, but are like important diseases, you can go and find patient populations that are very rare at any individual cancer center. But once you start putting multiple cancer centers together, you can actually like build a model for them. So this is one of dozens of examples where it's like a rare disease where we only saw 119 patients in the history of the Fred Hutch that have this disease and this treatment. But if you start putting together more and more cancer centers, you actually start getting a patient population you can study. So this is an example of the kind of tool that gets built from doing this. So this is just an LLM that in a secure and governed way is interacting with that federated learning system. And this is a clinical fellow, Simona Decker, asking a question about 2 targeted therapies for lung cancer. This is writing the code that then is being sent to that federated learning system. It's analyzing the data across 1 million patients. It's finding the patients that have lung cancer. It's looking at the patients that have those 2 treatments, comparing them, creating survival curves. And then she says, I don't really like the way that KM curve looks. Can I change the axis label? And it's like changing the axis label for her. So this is right now in like a prototype. There's lots of things we haven't solved with it yet that we still need to solve, like really who gets access to it, how do you govern how people use it, et cetera. But every clinical fellow in the United States would benefit from a tool like this because this is like 90% of what costs them time and effort in doing research automated and simplified with a tool like this. So these kind of resources have incredible potential impact. What we're doing with Kaya now is scaling it in 3 dimensions. More cancer centers want to join. We're adding more data types, and then we're adding more research projects. We have 8 right now, and we're going to be scaling up to many more over time. And so it's pretty exciting stuff. All right, so that was sort of, first it was sort of what we're doing at the Fred Hutch and why I think it's so important to create open resources. Then it's how do you do that at national scale? And then I wanted to kind of conclude with, you saw a little bit of an example of how this has a big impact on the world through this kind of Cancer AI Alliance tool that I'm showing you, but that was really a prototype. I wanted to just show you how in my own experience, these sorts of things can scale and can have really big impact beyond. And this is where I'm gonna talk for a minute about synthesized bio, so you can take everything I'm gonna talk about with a grain of salt. So one of the things that I just kind of believe and is such an important part of Bioconductor is that open infrastructure creates bigger opportunities. So recall, I worked on this recount project. It was a Bioconductor project. It started a long time ago. Based on that project, me and a colleague, Rob Bradley, who's here at the Fred Hutch as well, raised some money to start a company. And the company had this crazy idea. It said, in the future, most clinical data won't be generated from actual humans. It will be generated from models instead of from humans. So that was the crazy idea. And that idea is kind of, if you think about it, analogous to an LLM. I'm going to stick in some text. It's going to do something magical, and then it's going to print out the recipe that I asked it for. Here, it's like you're going to put in some text describing an experiment. It's going to do something magical, and it's going to print out the experiment that would have happened if you had done it in humans. And so like the big long-term goal is replace human clinical trials with virtual human experiments. That's a long way away, but this is the idea. And so the basic idea is you describe a collection of patients, you give them, characteristics of the treatments they have, their patient characteristics. You then generate at every tissue in their body genomic measurements or embeddings that represent those genomic measurements. And then you use those to do things like predict, are they gonna survive if you give them a certain treatment, even for new treatments. And so this idea is basically like building on what we did. Like GEM1 was released and was kind of based on just gene expression data. We're sort of expanding what data we're collecting with GEM2 and VHM1 and these kind of like virtual human models that we're kind of expanding on it. But all of this traces its roots back to an open data resource like Recount. And so what we did is we took Recount and we built on that idea, and we built a like constantly accruing set of data that comes from all of the public data resources and that like matches all the metadata and cleans all those data resources and makes them available for training. And what this lets you do is build these like much more complex models that cover many, many different data types. Again, using a lot of the architectures that like worked for large language models, but those architectures need to be adapted to genomics. Very famously, a bunch of these kind of AI models, like large virtual cell models, didn't beat linear models. And the reason that happened, in one big reason, is like they didn't model batch effects, which is like turns out to be kind of important in genomic data. And so it's equivalent to this famous problem. There's this famous problem in LLMs called the beep problem. I don't know if you've heard this problem, but when training your first LLM at a big scale, very often they don't converge. And there's this story where a group here in town, the Allen Institute for AI, was training their first a big LLM model, and it just wasn't converging. It wasn't converging. They couldn't figure out why. They went and looked at the data, the underlying data, and it found out that there was just like millions and millions, maybe billions of the word beep just over and over in the text data. And they were like, why is there so many beeps in the text data? It turns out there's a subreddit that is just people writing the word beep. many, many, many times. And so if you just download Reddit and you don't clean the data, you will have like so many beeps, it overwhelms all your other components in your model and it won't converge. Similarly in genomics, if you don't model batch effects, things are not going to go your way. So we've kind of built these models and made them available. We've then done a couple of interesting experiments with this. One is we regenerated all of TCGA, but with virtual humans, and then showed that they're very kind of closely replicate what's in TCGA. TCGA wasn't part of our training set. But really, the way we did this, we really first started building this model is we did something what we called reinforcement learning with Rob feedback. And so you can see the labels here, so this is not a very good experiment. But one of these are two heat maps of gene expression data. One is generated from a lab, one is generated from AI. And if you look at them, it's very hard to tell the difference between these two data sets. There's a couple of very small differences if you're really astute that you can kind of pick out. And in particular, you notice here that some of the variability is washed out in the GEM model versus more variability in the actual lab holdout data. But the way that we actually measure this data is generated in an area where my colleague Rob Bradley is an world expert. And so we would generate data and show it to Rob, like the AI data and the lab data, and we would have him play this game where he had to pick out which one was the AI data. And until he got bad at that game, we knew we weren't really onto something. So figuring out ways to evaluate these things is also really tricky. If you use those kind of latent variables or the generated gene expression data, you can put them into predictive models and then start predicting downstream things. And so one kind of cool recent example that we published is you can take one of these models and build it in, you can take one clinical study, build a predictor of whether you respond to an individual therapy, like an anti-PD-L1 or an anti-VEGF, and then you can use our model to essentially predict in a new clinical study how that combination therapy will work and who will respond to it and who won't. And you can do that with completely virtual trials. And so what I'm showing you here is two subtypes of lung cancer, and the responder rate predicted by purely virtual trials for the two subtypes is the GEM results. And then those are the actual results for different kinds of bispecifics, basically, that were from clinical studies that studied this exact sort of clinical response. You can then use that to sort of predict what might work in other tissues as well. Once you have this kind of general model, you can go and look at other diseases, and you can say, okay, if we know who's going to respond in lung cancer, can we also look and see what other diseases to prioritize? And so here, this is a graph of which diseases are most likely to respond to this bispecific therapy, and so you can kind of prioritize where you would expand into new indications. And so right now, this is being used by a bunch of people, biotech, pharma, finance groups, et cetera, to basically do a bunch of decision support. But ultimately, the goal is to make these models be more and more trustworthy so that they can be used in a more kind of direct manner to replace human studies. One of the things that's very cool about the way the world is working, I know I started with this idea that AI, it's not just LLMs, but mostly. And so one of the things that's so interesting is as you build any kind of tool, whether it's a bioconductor package or a large language model like the one we've just built, having them connect up to these kind of large language models enables kind of really cool things to happen. So here we've basically built a connector between Synthesized Bio and Claude. And so you can go connect to Synthesized Bio. If you have a Claude account, you can go look. It's in the connectors on the health connectors on Claude. And you can ask it to generate experiments for you. So you can have it generate gene expression differences in AML, comparing a couple of different treatments. And then Claude will write that into code that will then get sent to the synthesized models. It'll generate gene expression data from those models and write you a little report sort of comparing it. So you can kind of do these very specific experiments very quickly. The thing that's so exciting to me about this is not necessarily that application, though I'm very excited about that application. It's that there's this potential for all of us to have impact across all these different areas. And I think that there's the narrow view focus of AI, which is how do we use LLMs to accelerate X, or how do we use LLMs to accelerate Y? But I also think that there's this broader vision of how do we make data and computational resources available to everybody so they come up with net new ways of using AI that may or may not even include LLMs. Which I think is a very powerful opportunity across multiple different areas of drug development, patient health, generally as we define it. One of the things that is so critical, I think, is supportive, inclusive tech communities. These create outsized impact. And one of the reasons why I was so thrilled to be asked to give a talk here today is I think this is one of those communities. Having people come together, talk about these things together can have way more impact than you even think about it. And as an example of that, everything that I talked about here today, which is actually the work of many, many people, not just me, none of that would have happened if Bioconductor hadn't been around, because I basically wouldn't be a person that was like successful in my career without Bioconductor. And so like, it's really amazing how these communities can have impacts you don't even think they're gonna have. This is a very not comprehensive set of pictures of people who are involved. This is like all of the projects I told you about today probably involve on the order of 1,000 people across all the different projects, especially the Kaya one is enormous. So I wanted to say thank you to all these folks. And if your picture isn't up here, it's because there's enough in the room on the screen for me to thank all those people. And if there's anything stupid I said, that was my fault. Anything good is due to one of these people. So I think this is an awesome moment in time, both for the field. It's an awesome moment for Bioconductor. And if you're interested in any of the things I talked about here today, here's a bunch of websites where you can go check it out. And thank you very much for your time and attention.
All right, I think I'm the moderator for this one. And certainly I want to thank Jeff. I often get asked questions in introductory classes or whatever, what should I do? And one of the first things I say is, you need to make a friend. And I think we are really incredibly benefited by Jeff's friendship and his connection with the project, and I really want to thank him for this talk. Questions? I think I saw one over here, and you were the first one. Yeah, do you want to do a mic? I'll give you a mic. Yeah.
I think so the people online can hear. Oh, right, yeah. And if there are questions online, I don't know how we're going to manage that, but.
Yeah. Is this, people can hear me, right? Yeah, Kai work was incredible. And when I think about AI, one of the things I worry about the most is bias and how bias can be perpetuated.
Yeah.
And just, I think to me, it's not just the amount of data, it's also data composition.
Yeah.
So I'm wondering, like, how often do you think about bias with clinical models? And, for example, models trained in our community maybe may not generalize well to other communities. So just thoughts in general about bias and how you're thinking about that across your various efforts.
Yeah, that's such a great question. First of all, I'm going to say that I'm not the expert in this area. I know a lot of people who are experts, and I can point you at them, who are thinking about these problems very deeply, including on these projects. One thing I always think about is data can only represent the past, right? It's only, we collect it from the past, we store it, it's about the past. That means it bakes in everything that's good and bad about the past. And so every model you build, every way you think about this has to factor in the fact that like there is some baked in historical bias, whether that's good or bad. I mean, like there's some times where these models can be more effective because we've collected a bunch of data about a certain kind of problem, and that problem then we can answer very well. There's also really bad cases where it bakes in, biases against certain kinds of people and all that sort of thing. So every model should have some of that thinking involved. Kaya solves that a little bit in some ways, in that sense that by diversifying the populations we're looking at, it allows you to test whether things generalize. But as many people in the project would point out, we haven't really solved a lot of that problem because a lot of people don't actually go to one of these fancy comprehensive cancers. Like we've already biased ourselves to fancy comprehensive cancer centers. And so a lot of people don't get their cancer care there. And so how do you think about getting outside of that bubble? And then the other big thing is only certain data is collected when you go talk to your doctor, right? Like there's a bunch of stuff that you fill out on forms. There's some notes they put in. There's some labs they put in. But there's a gobs of information about your health that we just don't have in these data sets. And so that's a little bit more of a nuanced bias because it's sort of like you don't know what you don't know kind of bias. But yeah, so the answer to your question is yes, I and other people are thinking about it, me not as well as other people. And this is a super hard, ongoing, challenging problem. So if somebody wants to build a career, this would be an amazing area to figure out how to solve these problems because they're gonna be there for us for a long time.
Yeah, thank you.
Yeah.
We need more Kaya.
Yeah,
Appreciate it.
thanks. Yeah.
Hi, I was really excited to see your results about predicting trial results for the adenocarcinoma and the squamous cell carcinoma. And I was just curious, since clinical trials are the barrier for developing drugs, like, is there a reason, well, there must be a reason you can't instantly screen hundreds or thousands of candidates that are going to be, you showed like 50, 60, 70% efficacy. Like, you can't quickly find a candidate that's like 90% effective based on your predictions. Like, can you talk about that a little bit, maybe?
Yeah, so that's a great question. Yes, I would say that those models are currently being used actively by multiple different organizations to try to do that for specific diseases. What is, I think, still a bit of a gap for those models, and we're working on like kind of the next phase of that company, is genomic data is pretty well linked to the descriptions of the patients, and you actually do have pretty good descriptions of some of the therapies that they get. What's really, there's a real gap between genomic measurements and clinical outcomes. Like, we have it in subsets of cases. We have like subsets where those two things are linked. Some companies in some specific areas have that linked. Like, if you think about something like Tempus or Caris, they have some clinical data and a lot of genomic data. But nobody has really built a comprehensive resource that connects those two things together. And that's part of why those models, it's harder to do that kind of like global screening yet until we get a bigger resource that links clinical outcome data to genomic data. So that's kind of the next step for that company. But like, it's already being kind of tried out in those ways. And I would say it works okay. It will work better as we feed it more data. But yeah, the goal with those is, clinical trials, recruiting people to clinical trials is very hard. It's only getting harder. They take a long time. They're very expensive. To the extent we can optimize them in the short term and replace them in the long term, that's a huge opportunity for these kinds of models. But to do that, first you have to build trust that those models work, because you're not going to like go make that decision tomorrow unless you're really confident those models work. So that's a trust building exercise. Great question though.
Hi, so I have a question about the simulated experiments, or the hypothetical simulated experiments down the road. So, I mean, we run an experiment to answer a scientific question, and say you've built this model and it's powerful enough that it can just accurately simulate an experiment for you. Wouldn't the embeddings of that model also have the ability to just sort of answer the scientific question without simulating the experiment first, or is the simulation of the experiment sort of the proof that it's good enough? I guess my maybe very naive question is, why simulate an experiment when you can just get the answer?
I don't think that's a naive question at all. We get asked that a lot, and I think it's the right question. My experience with these things is often the embeddings do give you enough information. Like, if you tell an LLM, generate this virtual experiment, just give me the embeddings and answer this question, you'll often get a reasonable answer. My impression, though, in looking at these embeddings is like they're totally uninterpretable. Like, I don't know what that embedding is doing. Like, it's like super complex, super high dimensional. It loads a little bit on certain characteristics, but it's sort of like a PCA plot. You're not really knowing what's happening with these embeddings. And so I have found that the simulated experiments help the humans understand why the embeddings did what they did when they're answering that question. So often the embeddings is the best path. You're actually right, that's the best path to the answer. But the best path to get the human to understand why that's the answer is often through the simulated experiment. I mean, maybe in the future that won't be necessary, but right now I feel like that's the only way I can understand it. Like,
yeah, that makes sense.
That's a great question, though.
Okay, this is on. First off, very nice work. I'm very excited about like all the potential kinds of questions we could answer with like all the combined clinical data.
Yeah.
So one of the areas I'm particularly curious to get your thoughts on is oftentimes it has not been so much an issue of writing a model, writing the code for a model, but it's been more on the side of interpretation.
Yeah.
So you had mentioned as far as like potentially having clinical fellows coming in and be able to answer questions and deep dive the data and be able to get information out. So given clinical research already has a lot of issues with replicability of kind of like cookie cutter approaches where the individuals don't actually understand what's going into the process, how do you envision as we start building these systems, how do we actually try to make sure we're not just getting things that look convincing to us, but are actually getting at the underlying mechanisms?
It's such a great question. I would say that there's not an easy answer to that question. So it's good a question in that sense. Two things. One is the reason why I think things like Kaya are so important is if I build a model here, one of the best ways to know if it's a good model is to check it someplace else. Like even if I like am the best analyst in the world, and I like to think some of the students I've worked with are the best analysts in the entire world, or myself. I'll analyze some data, I feel like I did a great job, and then I actually try to reproduce it someplace else in some other data set, and it like doesn't work, or it didn't work because I didn't factor in some variable or whatever. So my feeling is, first way to solve some of that challenge of replication crisis and reproducibility crisis is to have places where we can replicate things, which is why I think Kaya is so important. The more subtle question that I think you're asking is like, how do we know, even if it replicates, that that's right? That it's not just some confounder or something that's causing that problem. And my intuition is that the best people to answer this question are people who understand the clinical biology, who have some training in epidemiology, biostatistics at the level of like what's a confounder, et cetera. So they like know what questions to ask when they're doing this, and then they can answer those questions. So I think like a combination of training and systems can go a long way there. Is this going to solve the reproducibility replication crisis in science? Absolutely not. Like this is like not, that's not what the goal is, but I think it is a great question. So.
So it was a beautiful talk. You touched on a number of things that strike me as fairly fundamental. Some of the questions did as well. The data is sampling from the past and generating a posterior that projects into the future. But meanwhile, new drugs are being synthesized, new trials are being schemed, new mechanisms are being discovered. And all of this is occurring against a background where the well-funded cancer centers are providing a lot of the data. But I once had a paper that went in and the referees all said, well, this is terrible, but you have to publish it anyways because you're never going to see a data set like this from Memorial or MD Anderson. And it was a bunch of patients from Rush in Chicago who were overwhelmingly younger African-American men who had APC wild-type colorectal cancer, which is supposed to be impossible. And so if the models are all trained against MD Anderson and Memorial Sloan Kettering in places where people can just buy an RV to go camp out and get their treatment, that ignores a huge swath of the world. And there's another one recently where we partnered up with hospitals in Pakistan to try and get sampling of a completely different population from what's going on in the United States or Western Europe. And I worry intensely that we've got these tech bro models and the anti-DEI initiatives in funding. And I wonder, is this the end for academic science? Is the only way this is ever going forward going to be if it makes money for somebody somewhere?
Well, I hope not. You have now gone above my pay grade, but I will do my best to answer what I think are like kind of a couple of fundamental questions that I think about a lot that are very similar to yours. One is the point that you made about Rush and the patient populations that we're including here. When we built the first prototype, you kind of had to go to places that were already prepared to do this because you couldn't do this fast with places that didn't have data infrastructure already. Now that the prototype is built, it makes a lot of sense for us to be very judicious about who do we add next. Is it 10 more fancy cancer centers, or is it places that represent the population? And so we've been trying to be very careful about how do we select who goes next. In terms of international efforts, there's multiple different international federated learning efforts, and we're like having conversations about how we have network effects so we can kind of connect us to bigger networks. The challenge with those is how many lawyers do I want to spend time with, because the sort of international regulations around data are very complicated and very different country by country. I once flew to Iceland to analyze a data set because it couldn't leave Iceland. So there's some complications to that, but I think that's the direction we're going. I certainly am a believer that in the long run, the investments that the US and the places around the world make in fundamental research that represents all of our people will ultimately turn into the kinds of things that, like, you can't have all of this innovation without these investments. Like, you don't get all these AI companies without open resources that let people build the first models, et cetera. And so I think these are actually mutually reinforcing ideas. I'm unqualified to talk about the politics, but I will say that I think that the right choice for our country and for lots of other countries is to continue to invest in academic science that represents everybody. And I think that's probably going to win out in the long run. But that's my belief.
On that note, I think I have to draw the meeting to a close, or this session
it
to a
down.
close, because we are over time, and Jeff has given a really tremendous overview, great questions. I really appreciate this. I think it's a wonderful way to begin the meeting, but we do have to draw boundaries.
Sounds good. Thanks, Vince.
Thank
Thanks,
you
Vince.
very much.